All right, so today we're going to go over transcription. All right, so transcription is a part of a two-step process that DNA goes through called protein synthesis. So what this means is we use the code that's in DNA to build proteins. All right, so that code that's in the bases that are in DNA, basically what that tells us to do is how to build proteins. Proteins are built by linking together amino acids, and it's going to tell us which amino acids to link together in the proper order to help us make proteins. All right, so transcription is the first part of this. It is the first part called transcription. And then the second part is called translation. So in general, transcription is taking a DNA code and converting it to an RNA code. And we'll go over why that needs to happen here in a little bit. Translation is then taking that RNA code and building a protein from that. So we can't just go directly from the DNA code into a protein. We first have to change it into an RNA code and then go into a protein. Okay? So we're just going to take a look today at transcription. How do we go from that DNA code to an RNA code? Okay? So first things first, all right? So I have to tell you a little bit about why this happens, all right? Why does transcription happen? We'll talk a little bit about RNA, all right? Then I'll show you that process because you're going to diagram it. So first of all, since we go from a DNA code to RNA code, this occurs in the nucleus. DNA is always in the nucleus. We do not want DNA to leave the nucleus because it can get damaged. So we want to keep DNA safe inside the nucleus. So anytime you're talking about a process that occurs with DNA, it has to occur in the nucleus. So if you remember, proteins are made at the ribosomes, out in the cytoplasm or attached to the endoplasmic reticulum. We don't want to take the DNA code and send it out to the ribosomes because it could get damaged. That DNA always stays in the nucleus where it's safe. So we convert it to an RNA code. The RNA can leave the nucleus and go out to the ribosomes to direct how to build a protein where the DNA is still safe. I say it's like if I had a blueprint to build a building. I don't send my original blueprint to the job site to build the building because it could get damaged or lost, and then I'm done. I don't have my original blueprint anymore. What I'm going to do is I'm going to make a copy of the blueprint and send that to the job site. That's essentially what transcription does. It makes a copy of the DNA, sends that out to the ribosomes to be able to build a protein. We're keeping the original DNA, the original blueprint, back in the nucleus where it's safe. Just like I keep my blueprint back at the home office instead of sending it out to the job site. Okay? So this occurs in the nucleus. It occurs so that DNA stays in the nucleus but sends a copy of the code to the ribosomes where we're going to build a protein. All right, so we want to keep that DNA safe, so let's make a copy of it into RNA and then take that to the ribosomes. All right, so let me look at a couple things first. All right. quickly. All right, so couple differences first between DNA and RNA, all right, before I get to how this occurs. All right, so we know that RNA is, obviously called RNA, so there's something different about it than DNA, all right, which I'll get to here. So let's talk about what's different about it. So RNA, oops. First of all, it stands for ribonucleic acid. So there's some differences between DNA and RNA, okay? First of all, it's called ribonucleic acid. It contains a different sugar that's in DNA. DNA, the sugar is called deoxyribose, but here in RNA, the sugar is just called ribose, which is why it's just called RNA, because it has ribose instead of deoxyribose. So if I were to build that sugar, all right, so remember this was deoxyribose in DNA? Well, this is called ribose. It still has a phosphate, and then it still has bases that are attached, but bases are different. The phosphate doesn't change. That's still the same, right? Phosphate. Here's the bases. The bases are different in RNA, slightly. There is still A, adenine. There is still G, guanine. There is still C, cytosine. All right, those are the same. So these are the same, but a base called uracil replaces thymine. So instead of there being a T, like there was in DNA, there's going to be a U instead. So it's got one base difference, and the sugar is different. And the other difference is that RNA is only ever single-stranded. DNA is double-stranded, double helix. RNA is single-stranded. There's only ever one side of RNA. All right, so you're going to see that here in a little bit. So over here, remember in DNA, A bonded to T. Well, now A is going to bond to U because uracil replaces it. G and C are still going to bond together every time. That doesn't change. But now we're going to have A bond to U. All right, so this is why transcription occurs. We need to keep that code in the nucleus, but we need to send a copy of that code out to the ribosomes, and that copy is going to be made by RNA, and then it's going to go out to the ribosomes. All right, so let's take a look at how this occurs. So the first thing that happens is, this is my DNA. And so what I did here was I split apart my two sides of DNA, right? So remember there's two sides. Think of this zip together, right? It makes the ladder. I unzipped the ladder right down the middle. So in between the A and the T, I unzipped it and pulled it apart. So this is going to code to tell us how to build a protein, right? So one side of this is going to code and tell us how to build a protein. This might tell us how to build a different protein, but this is the protein we need to build. So there's a code here in these bases that tells us how to build a protein, which we'll get to. But right now, we're just going to make a copy of this code to send out to the ribosomes. So what's going to happen is it will open up like this, and A will bond to U. So I'm going to write this underneath here. This is U. And what I did is I still made it like this. See, this T is like a darker gray. I made the uracil just a lighter gray because it's pretty similar to T. It just changes a little bit. G still bonds to C. That does not change. T still bonds to A. There's still adenine. C bonds to G. G bonds to C. And T bonds to A. Okay? So I have one base difference here. And then I have the sugar here is going to be ribose. So notice I have it red because this is deoxyribose in DNA. This is RNA. So my ribose, my sugar is going to be different. And then I still have phosphates in between there. Okay, so what's going to happen is, this is a sequence, it's going to code for a protein. So RNA comes here and bonds to it, and then what happens is RNA then is going to unzip from the DNA and leave and go out to the ribosomes, and this DNA is going to zip back together. So it opens up so that we can make a copy of one of these sides. This whole strand of RNA here, see, it's only single-stranded. It's going to leave, so like right here, it's going to cut off then, and this whole strand will leave and go out to the ribosomes, and this DNA, these two sides of DNA will get back together. This type of RNA that bonds to the DNA is called mRNA, and that stands for messenger RNA, because it takes the message that's in DNA out to the ribosomes. RNA can leave the nucleus. DNA stays in the nucleus. We don't want it to get damaged. But RNA is just a copy, and it's going to go out to the ribosomes, and we're going to read the code that's in there at the ribosomes to tell us which amino acids to put in which order. So that's transcription. I want you to diagram it. Here's what I want you to do. I want you to go back to your original DNA that you drew on Monday. I want you to show the two sides apart from each other like this. You pick one of the sides. I don't care which one. You are going to build an mRNA strand that bonds to one of your sides. So it will be 9 bases long. I want you to make sure that you label your new ribose sugar instead of the DNA sugar. And I want you to see that there's a uracil somewhere instead of the thymine. So those will be new things that you have to add to your key. And I want to see this strand just bonded to this. If you then want to draw a second one where the DNA gets back together and you show this mRNA out to the side, fine. But if this is all you do, showing the mRNA bonded to one of the sides of your DNA, that's it. That's what I want you to do. Label that transcription and upload it to the transcription diagram. All right, so you see how this mRNA is only one-sided. I don't have two sides of RNA bonded to each other. That RNA only bonds to one side of the DNA. So you're not going to draw an RNA over here, only on one side. I could draw an RNA over here, but then I wouldn't draw one here. I don't care which side you bond it to. Either one works. All right, so this is what your picture should look like. All right, but just know that then when you're done, when it's done coding for whichever sequence we need, this RNA will leave as a group and go out to the ribosomes, and this DNA will zip back together. All right, so that's the process of transcription. So it involves a type of RNA that's a little bit different, has a different base, a different sugar, all right, and it's only single-sided. All right, and it's the messenger that takes the message or the code that's in DNA out to the ribosomes. All right, so you're going to diagram this on your own again, just like we did with DNA. All right, everyone does their own. Okay, and then we'll talk about translation tomorrow, the second step of it. So what happens to this once it gets out to the ribosomes? We'll get to that tomorrow. Okay, all right, so that's your job for today, is to diagram transcription.